Hello, class. My name is Servan, and today I will discuss your protein metabolism. And I would like to congratulate you for reaching the last topic in your biochemistry lecture. And this topic is under your SAS number 10. Okay. We divided your protein metabolism into two parts. Here in part 1 class, we will discuss the digestion and absorption of your proteins. Okay, so we will discuss here how your dietary proteins can be converted into amino acids. And next, we have amino acid utilization. Here we will discuss what is the use of your amino acids in the body. Okay, please recall your SAS number 3, your proteins. You have different amino acids there, and they have different functions as well. And next, we have transamination and oxidative deamination, the production of your ammonium ion, and last, we have your urea cycle. Now let's discuss your protein metabolism. Okay, class, your protein metabolism is a crucial process in maintaining good health and is essential for various physiological functions in the body. We have here the importance of your protein metabolism, building and repairing tissues, and repairing tissues. What else? We have your enzyme function. This is still under your protein metabolism. Okay, we have your immune system function. And hormone regulation. Okay. Okay. So that is the importance of your protein metabolism. And please take note, class, that your protein metabolism or your proteins are also converted into amino acid. Okay, let me put it here. They are converted into your amino acids. And your amino acids, class, they have different functions in your body, right? Okay, so let me give you an example lang, like your tyrosine. I hope you can still remember the function of your tyrosine. Okay, let me put it here. Your tyrosine. What is the function of your tyrosine again? Okay, your tyrosine promotes healthy thyroid functioning. Promotes healthy thyroid. Let me give you another example. We have your proline. What is the function or what is the importance of your proline? So your proline plays role in your intracellular signaling. There. You can take a screenshot, class. And next, when we talk about proteins, class, let's not forget about the sources or where can we obtain your proteins or where can we get your proteins. Diba sa diet, okay? Ato ang mga foods. We have examples here. Let me erase lang this one. Sources of your proteins. Let me give you some examples lang, class, ha? So we have here your eggs, your meat, dairy product, and also your beans. There. So you have a lot of sources of your proteins, but these are some examples. Now let's proceed to your protein digestion and absorption. The first step is in your mouth. Just remember, class, that your saliva inside your mouth has no effect on digestion. The first step is in your mouth. You will eat your protein sources. You will chew it inside your mouth or you will grind it using your teeth. But Again, your saliva has no effect on digestion. So your protein digestion begins in your stomach rather than in your mouth. Why? Because again, your saliva has no effect on digestion. So your protein digestion begins in the stomach. There. Okay. Next, after you chew your protein sources in your mouth glass, it will go directly into your stomach. And then your stomach will release a hormone. We call this your gastrin. Okay, let me zoom this part lang. The presence of gastrin in your stomach class, this will secrete your hydrochloric acid and your pepsin. Let me just highlight the two. The secretion of your hydrochloric acid class or your HCl. Okay, we have three different functions of your HCl in the stomach. One of this is its acidity. The acidity of your HCl will lead to the activation of your pepsinogen. And it will form now your pepsin. And then the function of these two into your stomach, you have your HCl, it denatures your protein inside your stomach, and then your pepsin, it hydrolyzes your peptide bonds inside the stomach. So in short class, your protein denaturation and your protein hydrolysis occurs in the stomach. Okay? There. In your stomach class, the hydrolysis of your peptide bonds by your pepsin, this will result into your large polypeptides. So here, the hydrolysis of your peptide bonds again by your pepsin, this will result into your large polypeptides. And these polypeptide class, they will pass through your small intestine, where your small intestine will stimulate the production of your hormone secretin. I'm just gonna put it here. Secretin. There. Again, your large polypeptide will pass through into your intestine, and then your intestine will stimulate the production of the hormone secretin. And this hormone class, your secretin, okay, this will stimulate the production of your bicarbonate ion. The function of your bicarbonate ion class, this will neutralize your HCl, making it a basic environment. Then the basic environment class, this will allow the production of your pancreatic digestive enzymes like your trypsin, chymotrypsin, carboxypeptidase, and aminopeptidase. Okay? The function of these enzymes class, they hydrolyze your peptide bonds. So they attack your peptide bonds. And then after they will attack your peptide bonds, this will result into your amino acids. Then the absorption of your amino acid class through the intestinal wall requires the active transport with the expenditure of your energy. Okay. Here. Then after passing through the intestinal wall class, your free amino acids will enter the bloodstream. Then it will distribute throughout the body. There. So that is your protein digestion and absorption. Let's now proceed to your amino acid utilization. In this topic class, you will know how amino acids are used inside the body. Remember, we have different functions of your amino acid. So the question is, how do we utilize your amino acids inside our body? Okay, under your amino acid utilization, we have this term called amino acid pool. This is just the total supply of free amino acids available for the use in the human body. Okay, in simple terms, class, this is just the summary or how much amino acids are available to be used inside our body. Okay, now let's proceed to your sources of amino acid pool. We have three: dietary protein, protein turnover, and your biosynthesis of your amino acids. Okay, first we have your dietary protein. Where do we get this, or what are the sources of your dietary proteins? These are your meats, your eggs, and beans. Okay, I'm just gonna give three examples, class, but we have lots of examples of your dietary protein or your protein sources in your foods. Okay, now let's proceed to protein turnover. This is the second source of your amino acid pool, and this is the process in which your proteins are degraded and resynthesized. Degraded means that your proteins are broken down into parts inside your body, and resynthesized, your proteins are absorbed again or reabsorbed inside the body to be used again inside the body. Okay, now let's proceed to the biosynthesis of amino acid. This is the third source of your amino acid pool. Okay, but only the non-essential amino acids are biosynthesized because, as you recall, your non-essential amino acids class, they are amino acids that can be synthesized inside the body. Unlike your essential amino acids, these are amino acids that we can obtain from your dietary sources or your protein sources in foods. So now let's proceed to your nitrogen balance. What is nitrogen balance? This is the state that results when the amount of nitrogen taken into the human body as proteins equals to the amount of nitrogen excreted from the body in waste materials. Okay, in simple terms, class, this is nitrogen intake equals to your nitrogen excretion. Then we have two types of nitrogen balance. We have negative nitrogen balance and positive nitrogen balance. In your negative nitrogen balance class, this is the amount of your nitrogen in the urine that you excreted exceeds the amount of nitrogen that you ingested. In simple terms, class, your nitrogen excretion is more than your nitrogen intake. Then we have your positive nitrogen balance. This is your nitrogen intake exceeds your nitrogen excretion. Let us now proceed to the four ways to use the amino acids from the body's amino acid pool. Okay, we have four: your protein synthesis, synthesis of non-protein nitrogen containing compounds, synthesis of non-essential amino acid, and the production of energy. Okay, we will discuss them one by one. First, in protein synthesis, why do we need amino acids in protein synthesis? You need to remember, class, that you need amino acids to build the proteins, to perform important functions inside the body. Okay? You need your proteins to make components like your hormones inside the body, your enzymes, new muscles. So that is the purpose of your proteins class. And they can also replace and build new tissues in the body. So that's why your amino acids are very important in your protein synthesis. Let's proceed to the synthesis of non-protein nitrogen-containing compounds. Your amino acids are important in this part because your amino acids are also used in your DNA and in your RNA, your bases like your purines and your pyrimidines. If you remember, we have bases in your DNA and then your RNA. We have your A, C, T, and G, and also uracil class. Your amino acids are also synthesized to make heme. This can be found in your hemoglobin and also your hormones and neurotransmitters. So these are your non-protein nitrogen containing compounds. Next, we can also use your amino acids in the synthesis of non-essential amino acids. These are amino acids class that we can synthesize inside the body. Examples are your cysteine, alanine, aspargine, aspartic acid, glutamic acid, your tyrosine, serine, glutamine, and glycine. And lastly, we have your proline. And lastly, we can use your amino acids to produce energy. Okay, let's use this slide here. You need to remember, class, that your excess amino acids, they can't be stored inside the body. So the next step is to degrade them. Again, they are not stored, so they are degraded inside the body. Once degraded, class, your amino acids will result into two portions, your carbon portion and your nitrogen portion. In your carbon portion, class, okay, this will undergo further reactions to form compounds that can be used to synthesize your glucose and also your ketone bodies. And also, class, to synthesize your tags and ATP via your citric acid cycle. There. So how about your nitrogen portion, class, of your amino acids? Your nitrogen portion will be excreted in the urine in the form of urea, and also this can be used as a biosynthesis for non-essential amino acids and also biosynthesis of your non-protein nitrogen containing compounds. And so that's how your amino acids can be used in the production of your energy. Now, let's proceed to transamination and oxidative deamination. When we say transamination, this is the transfer of amino group. And then your oxidative deamination, this is the release of ammonium ion by converting your amino acid to keto acid. Remember, class, when we degrade your amino acid, it will result into two portions. We have your carbon portion and your amino portion or your nitrogen. There are two reactions happen in your amino portion. We have transamination and oxidative deamination. So it's under your amino group. First, let's discuss your transamination reaction. This is a reaction that involves the interchange of the amino group of an alpha amino acid with the keto group of an alpha keto acid. Okay, let me just zoom this part. We have two examples here. Okay, your succinate and glutarate. If we want to convert your succinate into keto acid class, we will add keto group here, and we will call this as your oxaloacetate. Again, succinate to keto acid, we will add keto group. Then if we want to convert your succinate to amino acid, you will add amino group here, and then this will be called aspartate. The same with your glutarate class. If we want to convert your glutarate into keto acid, we will add keto group. Then we will call this as alpha ketoglutarate. And glutarate to amino acid, we will add amino group. Then we will call this glutamate. Here we have your transamination reaction. Okay, let me just write it here. Okay, we will discuss step by step on how we can interchange your amino acid to keto acid. We have here the two most encountered keto or amino acid pair in transamination reaction. We have alpha ketoglutarate and glutamate. We have also aspartate to oxaloacetate. For this reaction to occur, your transamination reaction, you need a reactant and a product. I'm just gonna write the reactant at this side and your product in this side. So the products will be your oxaloacetate and glutamate in this reaction. And of course, to start this reaction, we need an enzyme. Okay, the enzyme needed for the transamination reaction is aminotransferase. Please take note of that. Okay, let's recall the definition again of your transamination reaction. This is a reaction that interchange the amino group of an amino acid to your keto group of an keto acid. We have an amino group here in your aspartate. We will transfer it to your alpha ketoglutarate. And after we transfer your amino group to your alpha ketoglutarate, this creates your product glutamate. Again, your amino group is now here. Again, from the definition, this is interchangeable. Your alpha ketoglutarate, the keto group, will transfer to your aspartate. Okay? Because this is interchangeable. So your keto group will transfer to your aspartate. Then you will have your product oxaloacetate. You need to also take note that this reaction needs a coenzyme, and the coenzyme needed for transamination reaction is your pyridoxal phosphate. Okay, so this is the coenzyme. Let's now proceed to oxidative deamination. Okay, the definition, this is a biochemical reaction in which an alpha amino acid is converted into alpha keto acid with the release of ammonium ion. In this formula, we have your glutamate, okay, and then the enzyme glutamate dehydrogenase to create your product, alpha ketoglutarate. Again, your product in this reaction is your alpha ketoglutarate. You need to take note that in this reaction class, ammonium ion is produced or released. Okay, this is your NH4. And the ammonium ion that is produced in this reaction class is very toxic when it is accumulated inside the body. That's why it needs to be converted into urea via your urea cycle. We will discuss your urea cycle after this topic. Let's now proceed to urea cycle. Okay, class, you need to remember that this pathway is cyclic and the product of this pathway is your urea. And then we will use your ammonium ions and aspartate molecules as your nitrogen sources. Your urea class is produced from mitochondria of our liver cells, and then it is transported into our blood, and then it will be excreted in our urine. And as you may notice, class, you have your first compound here, your carbonyl phosphate, which enters the cycle. We will discuss this in the next slide. Okay, so we have here your carbonyl phosphate. You need to take note, class, that this is the fuel for your pathway. Okay, the first fuel in your pathway, and this is formed from your ammonium ion. This came from your oxidative deamination plus your carbon dioxide. Okay, This came from your Krebs cycle or your citric acid cycle plus water and 2 ATP molecules. So that's how your carbonyl phosphate are formed. You have here your formula. We have ammonium ion plus carbon dioxide plus water and plus 2 ATP. So this will form your carbonyl phosphate. And again, this is the fuel for your urea cycle. Let's now proceed to the steps in converting your carbonyl phosphate to urea. Again, your carbonyl phosphate class, we call this as your fuel or the first fuel in this cycle. Then we have series of steps here. Step 1 is transfer. Step 2 is condensation. Step 3, cleavage. And step 4 is hydrolysis. We will discuss them one by one. Remember that your urea cycle is a cyclic reaction, so it means that this is a series of reactions that occur sequentially. So your last step will meet your first step. Okay, in your first step class, you have your carbonyl group transfer. With the help of your carbonyl phosphate, your ornithine will convert into citrulline and it will release your PI. So what happens here, class? Why do we call this as your carbonyl group transfer? Okay, so you have here your last step. You have your ornithine, and with your fuel, your carbonyl phosphate, it has its carbonyl group. It will transfer its carbonyl group to ornithine. Okay, then this will convert or result to your citrulline plus your PI. So that's what happens here inside your mitochondrial matrix or your mitochondrial matrix. Again, your step 1 happens in your mitochondrial matrix. You have to take note, class, that your citrulline needs to be transported into cytosol where your condensation reaction will occur. Okay? In this step, aspartate will enter. This is your second fuel in this cycle. Your citrulline will be converted into argininosuccinate with the help of your enzyme argininosuccinate synthase, and this reaction is condensation. So what happens here, class? You have your citrulline here, okay, and then your second fuel, aspartate, with the use of ATP. Now your citrulline via condensation reaction will converted into your argininosuccinate. So that's your step 2, condensation. Let's proceed to step 3. We have the conversion of your argininosuccinate to arginine. Again, this is step 3. We call this as your cleavage. Then in this step, you will release your fumarate. Then this will proceed in your citric acid cycle where it will continue the reaction there. Here in this step, class, you have your argininosuccinate. And with the help of your enzyme argininosuccinate lyase, this will catalyze the cleavage of your argininosuccinate to your arginine and fumarate. But only the arginine class will be used in the urea cycle because your fumarate will be used in your Krebs cycle. So it will be transported into your Krebs to be used in the process. And if you remember, class, that your second fuel, your aspartate that we used in your step 2, this came from your transamination reaction, the topic that we discussed earlier. Let us now proceed to hydrolysis. This is your step 4. Your arginine is hydrolyzed to form your ornithine and your urea. Okay? The urea formed from the hydrolysis of arginine class will be excreted in the urine. Okay. So what will happen in your ornithine? Okay, the ornithine from the hydrolysis of your arginine will be transported back to the mitochondrial matrix. Then it will proceed again to the series of reactions in your urea cycle when you convert your carbonyl phosphate to your urea. So that's it for your urea cycle. You may now proceed to the part 2 of your SAS number 10. Thank you for listening.